Today we are going to study about isozymes. Uh, Isozymes are enzymes that catalyze the same reaction, but they are encoded by different genes. We can define them as the same reaction catalyzed by two or more different forms, molecular forms of an enzyme. These multiple molecular forms are called as isozymes. These multiple forms may occur in the same species, same tissue, or even in the same cell. These multiple molecular forms differ in their kinetic properties, in their regulatory properties, in their distribution pattern in the cell. They can be present in the cytoplasm, or they may be found in a certain cell organelle, or they can be present in soluble form or membrane bound form. They can differ in the type of cofactor that they are using. For example, in case of dehydrogenases, they may either use NADH or NADPH. Various isozymes may have similar but not identical amino acid sequence because they are encoded by different genes. For example, lactate dehydrogenase, LDH in vertebrate tissues exists in at least 5 molecular forms, and these forms can be separated by electrophoresis. All LDH isozymes contain 4 polypeptide chains. These polypeptide chains these polypeptide chains are present in different ratios, and each isozyme contains a different ratio of two kinds of polypeptides. The two kind of polypeptides are M chain. M stands for muscle, and the H chain, which H stands for heart. In skeletal muscle, the main isozyme form, which is present in greater amounts, is it contains 4 M chains, while in heart the main isozyme contains 4 H chains. Different LDH isozymic forms are found in different tissues, and they are formed by a combination of the two different type of polypeptide chains. LDH1, which consists of 4 H chains, is found in heart and erythrocyte. LDH2, which consists of 3 H and 1 M chain, is also found in heart and erythrocyte. LDH3 consists of 2H, 2M chains, brain and kidney. LDH4, 1H, 3M chains found in skeletal muscle and liver, and LDH5 is made up of 4M chains and is found in skeletal muscle and liver. The difference in isozyme content of different tissues help us to diagnose or help us to differentiate which tissue is diseased, because the isozymic pattern of each tissue is different, and it is generally used to assess the timing and extent of heart damage due to myocardial infarction. When damage to heart tissue results, it results in the release of heart LDH into blood. Initially, shortly after heart attack, the blood level of LDH, total LDH, increases, and at that time LDH2 is greater than in concentration than LDH1. After 12 hours, LDH1 and LDH2 become equal in concentration, and after 24 hours, LDH1 is more in concentration than LDH2. So this switch in the LDH1 to LDH2 ratio, combined with increased concentration of other in the heart enzymes like creatine kinase, it is a strong indication that a recent myocardial infarction has occurred in the patient. Different LDH isozymes have significantly different values of Vmax and Km, particularly for pyruvate. Properties of LDH4, they favor rapid reduction of very low concentration of pyruvate to lactate in skeletal muscle, whereas those of isozyme, while LDH1 favors rapid oxidation of lactate to pyruvate in heart. The distribution of different isozymes of a given enzyme reflects at least one of the 4 points. First is different metabolic patterns in different organs. For example, glycogen phosphorylase, the isozyme in skeletal muscle and that present in liver, they have different regulatory properties reflecting different roles of glycogen breakdown in in uh, in these two tissues. Then different locations and metabolic roles for isozymes in the same cell. For example, isocitrate dehydrogenase, it is found in cytosol and mitochondria. Third is different stages of development in embryonic stage or fetal tissues as well as in adult tissue. For example, fetal liver has a characteristic isozyme distribution of LDH, which changes as the organ develops into adult form. So adult form has a different LDH pattern than fetal form. Then important some enzymes of glucose catabolism, which are present in malignant tissues. They have a fetal pattern of isozyme rather than the adult pattern. Different fourth is different responses of isozymes to allosteric modulators. That this difference is useful in fine tuning of metabolic roles. For example, hexokinase 4, which is known as glucokinase of liver, and hexokinase isozyme of other tissues. They differ in their sensitivity to inhibition by so these isozymes, glucokinase and hexokinase, they differ in their sensitivity to inhibition by glucose 6-phosphate. Then for differential diagnosis of disease, isozyme pattern is studied. Different isozymes have different inhibitors and heat stability. For example, myocardial LDH, that is LDH1, it is heat stable as compared to hepatic LDH, which is LDH5. Hepatic LDH is also inhibited by urea. So these two properties help us differentiate between hepatic LDH and heart LDH. Whenever a person suffers from myocardial infarction, LDH1 and LDH2 increase, and we can come to know about their increase by studying their heat stability, while in liver diseases, especially uh, when a person suffers from acute viral hepatitis, LDH5 and LDH4 predominate. So by studying them, their urea inhibition, we can come to know about these isozymes, what is their content. In this way, differential diagnosis is made. Then isoenzymes of creatine phosphokinase, it occurs in 3 isozymic forms. Each isozyme is a dimer made of 2 subunits. One is M subunit and the other is B subunit. Different isozymic patterns have different type of subunit composition. CPK1 has BB found in brain, CPK2 MB myocardium, CPK3 skeletal muscles. Normally CK2 isozyme is very small, but after myocardial infarction its concentration increases very much, CK2 increases. Then isozymes of alkaline phosphatase, LP, exists in a number of isozymic forms. Main are hepatic isozyme, bone isozyme, placental isozyme, intestinal isozyme. Clinical significances, the major liver band increases in many hepatobiliary diseases or in liver diseases. Bone isozyme increases due to osteoblastic activity, which is present in bone cancer or diseases related to bone. Apart from it, normally it is elevated in children because of greater osteoblastic activity due to their growth. In pregnancy, placental isozyme of ALP increases. It is heat stable and inhibited by phenylalanine. Atypical alkaline phosphatase enzyme isozyme, Regan isozyme, it is produced by malignant tumors.